simple pieces to our genetic code, right? Cytosine, guanine, adenine, and thymine, C, G, A, and T. Well, not anymore, folks, not anymore. Now, there are 6. When you actually think about it, trying to make an entire complex being and all the crazy stuff that goes with us out of only 4 simple pieces isn't very efficient. The proteins that our DNA codes for are themselves made out of amino acids, of which there are only 20. Isn't that crazy? Every living thing is made out of different combinations of the same 20 basic building blocks, coded by just 4 even more basic building blocks. So it's safe to say scientists have been dreaming about expanding that toolkit for a long time. And that's right, we've upgraded. These unnatural additions to genetic code have names like this and this. But to simplify things, we call them X and Y. And this class of synthetic DNA base pairs are called xenonucleic acids, or XNA. Biochemists first started trying to add these new base pairs to DNA in the 1980s, which, as you can imagine, is not easy. Base pair bonding in natural DNA is highly accurate. A always bonds with T, and C always bonds with G. But the pairs we've added are a little harder to get right. It's easy for these newbies to mispair. This is because the hydrogen bonds that hold the 2 sides of a DNA helix together are really weak. And while they're enough for the natural stuff, holding synthetically introduced base pairs together properly requires something a little stronger. One team has gotten around this bonding problem by making the new base pairs stick together because they're both hydrophobic, making them line up properly to bond in an effective way. Others have simply tried to strengthen the hydrogen bonds by manipulating the structure of the unnatural bases. But that's really just the first hurdle. Once you've gotten the new base pairs to stick together, you have to make sure they don't disrupt the rest of the DNA and make chunks of it non-functional. And then you have to get the cells of whatever organism you're adding it to actually accept them, and to be equipped to produce whatever these new bases are asking them to produce, which seems like a pretty big headache. Nature is just doing fine on its own, a master technician with all its gorgeous cellular machinery, so why do we want to mess with it so much? Well, because it's worth it. We already genetically engineer organisms like E. coli to produce a desired chemical product for us. Because their genome is easy to infiltrate and they reproduce so fast, you can make industrial quantities of the thing you want, like insulin, for example. But as we've discussed, we're only working with 20 amino acids to build everything out of. Adding in synthetic base pairs could make it possible for organisms to be able to build proteins out of more unusual materials, like synthetically produced amino acids, maybe metal-binding ones that would be conductive or magnetic, making biomaterials that could be used in electronics. Okay, but aren't we getting into mutant superhero territory here? Well, it's easy to want to make that leap, but we're not quite there. Because all of these materials are synthetic, both the base pairs and any unnatural amino acids those base pairs would code an organism to use, they're obviously not found in nature. Scientists have to provide these materials to the organisms in order for the organisms to be able to use them, and in most cases, even to survive. If the modified organism doesn't have the materials to make these synthetic base pairs, they just die. They couldn't run rogue Jurassic Park style even if they wanted to. Realistically, we'll use these new capabilities to produce new catalysts, which are used to improve and speed up chemical experiments. And this could radically transform our ability to engineer and mass-produce more effective medicines. With new DNA and new amino acid building blocks, we have control over what proteins are produced at a level of detail we could never have previously imagined. It's like we've been trying to cobble together shoes by hand, but now we have access to a state-of-the-art shoemaking factory. We just have to figure out the blueprint of the place and the high-tech security system. And who knows, maybe the work that it takes to get us where we want to go in this field will help us better understand the lingering mysteries of DNA, like how it all came together at the beginning of everything to create life. For more on the science fiction-like reality of DNA manipulation, check out this video here. And as always, thanks for watching.
Genetically modified organisms you didn't know about. 18. Featherless chicken. Israeli scientists have created this strange animal, a chicken without any feathers. The weird-looking animal is supposedly much cheaper to raise, more environmentally friendly, and doesn't require any feather plucking, which, in general, saves resources. This naked chicken can literally go straight from farm to fridge without much work. While this seems like a great idea, there are still some hurdles to cross. such as the fact that, without feathers, chickens cannot protect themselves from harsh weather and parasites. 17. Glittering gold seahorses. Vietnam-based scientists have created a breed of seahorse that is literally worth its weight in gold. Scientists took the fluorescent gene from a jellyfish and inserted it into a tiny grain of gold, which was then injected into the eggs of a seahorse. This glittering gene was absorbed into the cells of the seahorse, and they grew into glittery seahorses that shine in the light. While it seems like a lucrative venture to sell glittery pet fish, the scientists were testing out a technique to replace negative genes with good genes in humans and livestock. 16. Singing mouse. While we usually genetically modify animals and conduct experiments with a direct purpose, the Evolved Mouse Project doesn't really do that. Instead, they add sort of random genes to mice, let them breed, and see what happens. That's how, after mixing mice genes with bird genes, we got singing mice. Baby mice were found inherently to know how to sing from a young age. When other non-genetically modified mice grew up around the singing mice, they actually learned how to make similar sing-song chirpy noises themselves. 15. Glowfish. There is a pet fish whose DNA is patented and trademarked, and that fish is the glowfish. They are genetically modified zebra daniofish that were crossed with the fluorescent proteins of a jellyfish to create fun fish that glow different neon colors. Other glowfish were created by splicing the zebra danio with sea coral and other jellyfish genes. You can own your very own GMO fish in most parts of the United States, except for California. 14. Spare part pigs. It's pretty common now to use the heart valves from pigs to transplant into human hearts. But now, we can develop pigs that we could take the entire organs from for human transplants. These pigs are used for spare parts and have been designed so that their hearts, livers, kidneys, pancreases, and other organs can be taken whole to be put into humans without our body's immune system rejecting it. 13. Fast-growing salmon. When it comes to sustainable food sources, we usually look towards domesticated animals and plants that we can farm and breed. Salmon is sometimes farmed, but they take a little while to reach maturity.
Even though we've been living on this planet for thousands of years, we're still a long way from discovering everything about it. Sometimes we find something that's been hidden or buried for hundreds or even thousands of years, and changes the way we think about our own history. Here are some truly incredible recent discoveries that have shed fresh light on the past. Those who go digging for buried treasure are used to finding most things in the ground, but Swiss archaeologists were more than a little surprised when they discovered this bronze hand. They asked other experts around the world what it might mean or represent, but none of them knew either. It's been carbon dated back to 2000 BC, but nothing like it from that time period has ever been found. It's too delicately constructed to have been a prosthetic, and nothing about the skeleton or the gold pieces and jewelry found buried with it offer any further clue at all. The best guess of archaeologists is that it's a symbolic replacement for a hand the buried man may have lost during life.